Well, good afternoon, everybody. Thanks for coming to the presentation, and special thanks to Tahiri for introducing and joining us on our trip this year. I won't go into the details, but if Tahiri steps out for more than 5 minutes, go help him. And he can explain that if he wants to later. So, yeah, today I'm going to present on the research we've been doing over the past few years here in Madagascar. This is my third trip and third talk here, and over time we've kind of been building our data on this disease that initially we were working on in Florida, and it was discovered here in Madagascar. A similar strain was discovered in Madagascar, and I'm kind of going to do a little bit of background on what we have in Florida and how and why I made it here to study this disease here in Madagascar. So, palm lethal decline phytoplasmas, it's a type of plant pathogen that is essentially, it's a type of bacteria. And there's a lot of different species that affect different agricultural crops. And in a lot of herbaceous hosts like strawberries, tomato, things like that, they cause some unusual symptoms, but they're not lethal to the plant. This group of phytoplasmas is actually lethal to the palms that it infects. And as far as we know, there's no palm that is resistant to the infection. So pretty much once a palm is infected with the pathogen, it dies, and the disease cycle kind of perpetuates depending on whether or not the vector is present. So the disease we work on is called lethal bronzing. And a little bit of background, in the New World is where they really first categorized this group of plant pathogens. And the first disease to be documented was in Jamaica in like the late 1800s, and they called it lethal yellowing. And around 2006, a new strain of the pathogen showed up on the west coast of Florida around the Tampa area. And since that strain has been introduced to the state, it's become the most prevalent, it's displaced lethal yellowing. So we had lethal yellowing in the past that primarily affected coconut, excuse me. And when this disease got introduced in 2006, it has a slightly different host range. So it affects some of the native palms we have in Florida, but it also affects some of the ornamental palms that are more cold tolerant and have a wider distribution in the state. So this lethal bronzing has spread through most of the state of Florida, and we're starting to see it in nearby states just to the north. So the overall impact of this disease is going to be far greater than lethal yellowing, because lethal yellowing there was other hosts, but the primary host was coconut. And in Florida, it's really the southern 1/3 of the state that really has a tropical climate that we can grow coconuts. And then in the northern part of the state, it's a little too cold, so we don't have the primary host of lethal yellowing. So LY was kind of limited in its geographic spread, whereas lethal bronzing has a much larger geographic distribution. Now, at this point, the pathogen affects over 20 different species of palm, but palms in the genus Phoenix, and this is our native palm, sable palmetto, which is one of the primary hosts of the pathogen in the state. And here, I just wanted to show what the symptoms look like in a cabbage palm that's infected. And typically what happens is, once the vector transmits the pathogen, we believe the latent period is anywhere from 4 to 8 months. It might be a little bit longer in some circumstances. But once the symptoms appear, it's pretty much a death sentence. The plant's not going to survive. So there's currently no cure. So there's a few techniques we have to try and slow the spread, such as rapid identification and detection, so that we can remove infected plants sooner. But there's also antibiotics that you can inject to prevent the infection from getting hold. But this here just shows the symptom progression, and regardless of which phytoplasma in Florida or the New World, there's also a strain in Tanzania, and as I'll mention, there's a strain here in Madagascar. Regardless of which of these phytoplasmas you're talking about, and regardless of where, they're all lethal, and they all produce similar symptoms. And the first symptoms of a phytoplasma infection, if there's fruit or flowers present, is you get premature fruit drop or death of the flowers on the palm. Now, depending on the time of year and species, that may not always be a reliable symptom. But if those structures are present, those are going to be the first things to die on the plant. Following that, you start to see dying of the oldest fronds or the oldest leaves first. So the leaves that are closest to the ground on a palm are the most mature, and those are the ones you see dying first. And that death slowly moves further up into the canopy, and then around 50% canopy loss, the meristem, the main growing point of the plant, dies and the plant's dead at this point. There's quite a bit of variability in how long this symptom progression takes, which could be due to the overall health of the plant, environmental conditions, how many insects carrying the pathogen infected it. But I would say on average, from symptom onset to complete death, you're looking at like 3 to 4 months is like the average time frame. Sometimes I've seen them go faster, sometimes they maintain green foliage quite a bit longer. A few years ago, we identified this small plant hopper in the family Succiidae. It's a hemipteran insect, and we confirmed that it was the vector by doing some transmission experiments where we exposed the insect to the spear leaf of an infected palm, and we allowed it to feed for up to 5 days. And once it acquired the pathogen, we placed it on a cap with artificial feeding media and allowed it to feed there, and we used something called digital PCR, which I'll talk about a little more detail later. And we used this to detect it in the artificial media to confirm that this species was in fact the vector of the pathogen. On top of that, we did survey work, excuse me, over a year and a half where we collected specimens where there was disease spread, and the only species that consistently tested positive was this insect. Now, this has been the majority of our research program in Florida. And over the years, I became interested because there was other plant hoppers associated with palms. And we initiated some surveys in places like Costa Rica and Jamaica, where we were looking for other potential vector species that could transmit the pathogen. And when we started surveying, I was collecting plant hoppers from the palms, and I contacted a colleague of mine who's a taxonomist and specialist in plant hoppers, and I asked him what these were, and he had no idea. It turned out to be new species. So we started kind of increasing our survey efforts. We were still primarily focused on the phytoplasma and the pathogen and coming up with management strategies, but the kid in me that likes to collect insects and look for new bugs was curious about all these new plant hoppers we were finding on palms. So we increased our effort to just look at the overall biodiversity of plant hoppers on palms. And since we started that, we have like, I'm certain that we have over 200 new species of insect just from Costa Rica and Jamaica and in the New World alone. But there's clearly an interesting relationship between these insects and palms specifically. So over time, I was kind of curious if this relationship was universal, like if we would see it in the old world as well as the new world. There was some literature that suggested that there were plant hoppers, a lot of them on palms, but it seemed like an interesting area to explore. So when I was looking for places, Madagascar seemed like a good spot because of the high levels of biodiversity, but also the high levels of endemism of pretty much all flora and fauna here. So it seemed like a good region to focus on to look at this potential relationship in the old world. So I initiated the process to come here to start documenting plant hoppers on palms, but in the meantime, a publication came out that the phytoplasma disease, a similar strain was documented from northwestern Madagascar. So when that happened, it kind of gave me further justification to seek funding and support for coming here, not just for plant hopper diversity, but now we had a similar strain of this pathogen that I was already working on showing up here, so we could start exploring the vector relationship here as well. So, as I mentioned, it was in northwestern Madagascar. The first reports were from Ansui. This is the picture from the publication. And Katsepi is where they found the first cases of this phytoplasma infecting coconut palms. And you can see, similar to the pathogen we have in Florida, you get this heavy discoloration and death of the oldest fronds first, and that eventually is going to kill the palm. So when this report came out, I contacted the lead author, Fabien Pilet, and he works for the CIRAD with the French government. At the time, he was located in Reunion Island. So we started communicating and planning some trips together. And the last 2 years I've been here, he's joined me on these survey efforts to not only look for infected palms, but also document potential vectors on the plants themselves. Now, my first year here in 2023, we went to the eastern side of Madagascar just because it was my first trip here. I didn't know a lot of the places, and I just wanted to get a feel for collecting and what the diversity was. So we went to Mitsingo Reserve and Analalava just to see what was out there. We didn't find any infected palms, but we got a lot of plant hoppers. And actually, from 2023 survey, we got 64 species of Suxian and 42 species of Durban in just 2 families. So well over 100 species were collected on our first survey, but no phytoplasma. Last year, when I came back, we focused in the actual area where the disease had been originally documented from. And we settled on these 4 primary sites in Katsepi, Ansui, Mampikoni, and Ambondromami, I think. Sorry. But this shows the general areas where we collected. Now, when they first found the phytoplasma, they documented it in Katsepi and Ansui. So we went back to both of those sites, and then these other 2 sites were just, we were driving around and noticed some dying palms, so we stopped and surveyed the area, and they became quite productive. So those were our 4 primary sites that we focused on in our survey last year. We did go to some of the natural, like the national parks and private reserves, not for the phytoplasma, but just for general plant hopper collecting. So I'm not going to get into those details here. But these 4 sites were the focus of our vector work for the phytoplasma. And actually, excuse me, one of the best sites we had was this small little plot in Mempaconi that had a lot of Bismarck palms, and they were covered in this small succeed. And as soon as I saw that insect, I had one of those moments where I was like, that's our vector. I was fairly confident of it, but the rest of the trip, we had to collect and eventually do tests once we got back to Florida to confirm it. But as soon as I saw that insect, I was almost certain, because it's a somewhat close relative to Hyplaxias crudus, the vector of the phytoplasma in Florida. So this is one of our field sites. I don't have pictures of Katsepi. We didn't find any of these insects in Katsepi, and there was no sick coconut palms there, so I'm not going to include that really in the discussion because we didn't get anything. We got quite a few plant hoppers from this site in Ansui. We got quite a few of this succeed at this site there. And interestingly, the owner of this property had a coconut plot. And when we visited, he told us that it was a few years prior to us arriving, he had lost an entire field of coconuts. And he didn't know why. And I'm almost certain it's because of the phytoplasma disease. And in the area, in the general area, there's quite a few of this insect that I was, at the moment, pretty confident was going to be the vector of the pathogen. And then this was our final site. This was in a front yard, and I noticed this dying coconut palm from the road. So we went in and asked the owner if we could sample and collect in the area, and he let us, and we found more of the same insect on the coconut palm. So my confidence was growing that this was the bug responsible, and it was pretty exciting. Now, in the moment I was collecting these insects, and they all looked the same out in the field. So I thought I was collecting a single species. But as it turned out, there were 3 species that we were collecting, and I didn't realize it. The genus of plant hopper is Nizomindus, and in the literature there's a single species that was known previously, Nizomindus australis. And this species was historically known. I forget the national park that was south of where we were, but it was also documented from an island off the northwestern coast of Madagascar. So we were right in the middle of the range of this species. So when we were collecting it, I said, okay, we have Nizomindus australis. Then, I think it was like towards the end of the trip, I saw this one specimen that had this big black spot on the forewing. And it made me suspicious that there might be multiple species that we were collecting. So when I got back, I started doing the dissections, and it turned out that there was, technically there's 4 species, but I'll get into that a little bit. Of the specimens, we collected over 100 specimens. There were 3 species within this material. We collected the type species, Australis, but the 2 new species documented that I just finished writing the paper and will be submitting it soon, is describing one called Madagascarensis and one species called Macula because of the spot on the hind wing. The tricky thing is once we started doing the molecular analyses, we realized that this spot on the forewing is not consistent. So a lot of specimens that we're sequencing and matched my type specimen of macula for the CO1 gene, a lot of them didn't have that spot on the wing. Now that could just be an age-related thing, a developmental thing. We're not sure. That feature is not a reliable diagnostic character for the species. I'll show some morphology here in a minute to show the actual difference. But you can see why superficially in the field when I was collecting, I just called them all the same species. And it wasn't until I got home and sorted them and dissected that I realized we had 3 distinct species. So once I got all the species sorted, we did some molecular work to kind of supplement our species description. And this phylogenetic tree is based off of the CO1 gene, both the 5' and 3' half. The H3, histone 3, it's a nuclear gene. CO1's a mitochondrial gene. 18S ribosomal RNA gene, and 3 different expansion regions from the 28S ribosomal RNA gene. And this is based off of a combined analysis of all those markers. And you can see, I know the numbers aren't great, but it's 100. So there's very strong support for for Nizomindus being a monophyletic group. But interesting, it's a very long branch that does not resolve well with the other succeids that are more closely related to the vector that we have in the New World. But long branches in phylogeny seems to be the norm rather than the exception with insects from Madagascar. I don't know why, but a lot of the bugs I get from here, they have very long branches. So it's kind of interesting, but you can see here there's good support for these 3 species, even though they're superficially identical, for more or less, they're genetically distinct. Now, the interesting thing was when we went through and barcoded, and we used the CO1 gene because it can be a useful population genetic marker if you don't have microsatellites, markers to perform analyses. And what we found, so this group here is Nizomindus australis, the type species. This one here is Madagascarensis, and this big clade up here is macula. So macula was the most abundant species we encountered. And it turned out, we have to do the formal analysis, but it seems that macula prefers the disturbed urban settings, whereas Madagascarensis, and australis, we seem to find that more often in the natural ecosystems. Another interesting factor, it's kind of hard to see because I have so many samples, but there are multiple genetic haplotypes within each of these populations. So there's something very interesting evolutionarily going on with plant hoppers here, because not only are they on a long branch, but you have a case of what appears to be sympatric speciation, because we found, even though there's preference for habitat, we found all of these species in the same general area. So in the natural areas, we did find some macula, but australis and Madagascarensis, and then in some of the urban areas, we also got australis and Madagascarensis, not as common, but they were still there. But on top of that, you have high levels of haplotype divergence, which initially I was concerned that it was PCR error because you're talking about a few nucleotide differences. But we resequenced everything and basically got the same sequences and the same results. So there's some really interesting things going on genetically with this group of plant hoppers. And this branch down here is a single female I collected. It's a different species. but for the moment I can't do anything with it because when you describe plant hoppers, you really need the male, because the male morphologic characters are the most informative, and most of the literature is based on that. So we did collect some Neosomindus on this trip, and I'm hoping I got the male this time, but we'll see. And we have our out group of other plant hopper species in the tribe that is supposedly the same tribe as Neosomindus. Here, this is the male reproductive organ, and this is the primary structure to delineate species in plant hoppers. So this here is Australis, and you can see this big concavity here on the posterior margin. This is Madagascarensis, and there's a huge difference between Australis and Madagascarensis and macula. The difference between these two is much more subtle, but at least at this structure, it's consistent. If you remember at the spot, it's not a good diagnostic feature, but here you get a slight concavity on the margin, and these two spines are turned upward. In macula, it's convex, and the spines are curved downward. So it's a small difference, but it's consistent. And with the molecular data we have, it's good support for establishing these two new species. Now, this is from the phylogenetic standpoint of just discovering these new plant hoppers, excuse me, on palms. Now, the next step of this was understanding the relationship of these plant hoppers as it pertained to the phytoplasma disease. As I mentioned, I was confident that this species, a single one, which turned out to be 3, was the vector. So basically, we went through all of our specimens, whether they were found in natural areas, on healthy palms, or in the city areas on sick palms. And basically, we did dissections of the salivary glands. So this here, you can see this kind of nebulous white stuff. Those are the salivary glands of these plant hoppers. And it's a little tedious because they're actually situated in the prothorax. So they kind of get connected, and if you just remove the head, the salivary glands will get stuck. So you have to take the prothorax and the head off together, and then remove the prothorax from the head, and then basically scoop out the salivary glands from the back of the head, and we do the DNA extraction on that and test those for the presence of phytoplasma. Because phytoplasmas in general are spread in usually a propagative manner. So what happens is the phytoplasma is ingested by the insect, it traverses the gut wall, enters the hemocoel, migrates to the salivary glands, enters the salivary glands, and then the insect can inject it into a new host plant. So generally, if you find pathogen in the salivary glands of an organism, that's an indicator that that species can transmit the pathogen. So we went through all of our specimens and scooped out the salivary glands and tested them by digital PCR. And this chip image is kind of the visual representation of digital PCR results from salivary glands. Excuse me. I guess a brief explanation of digital PCR. It's a highly sensitive protocol that's approximately 100 times more sensitive than qPCR, at least in our system with some of our tests. And what happens is you take a sample and you load it into a plate, and then the machine pushes your sample, your reaction mixture, like you would a normal PCR reaction, and it separates it into 20,000 very small PCR wells. And it allows for the detection of a single, so each one of these blue pixels functionally represents a single target copy of your DNA. So it allows for absolute quantification of whatever it is you're detecting. If you do something like quantitative PCR, you do estimates on DNA of established concentrations, and you do a serial dilution, and basically the software estimates your sample based on the strength of the reaction relative to the standard curve of DNA at known concentrations, and gives you an approximation. So with digital PCR, it's much more accurate and much more precise, and much more sensitive than quantitative PCR. And when you're dealing with looking for trace amounts of DNA in salivary glands, or like I mentioned with the artificial feeding media for the transmission assays, you need that high level of sensitivity. So we went through all of our specimens and dissected salivary glands and extracted the DNA and tested them all by digital PCR, and then we went back and tested the bodies by standard PCR to try and get sequences to confirm, to correlate with the salivary gland data. And what we found is most of the specimens were negative from most of our sites, but about 7 specimens from our site in Mampocony tested positive for the phytoplasma. And we got, not all of them had them in the salivary glands. Depending on where the pathogen is in the body, sometimes you can detect it in the body, but it hasn't made it to the salivary glands yet. But we had 7 specimens that, whether it was salivary glands or the body or both, tested positive for the phytoplasma. And interestingly, it was only that species, Nisomindis macula, that tested positive. Now at this site, that was the only species we found. So it's possible that the other 2 species, if they were in the presence of the pathogen, may be able to acquire it. Now at this site, we got 1 specimen of Australis macula and Madagascarensis, and we had a palm that was dying of the infection, but none of the specimens tested positive, which could just be what happens in the infection is it takes time for the pathogen to invade the tissue where the vector's feeding. So given the stage of decline of that coconut palm, it's possible that the insect had just started feeding and the titer was not high enough in the specimens, and we just didn't get them. But we got good data from the specimens in Mampocony and detection in the salivary glands. So this pretty much told me that at least this one species, I think it's possible that the other 2 can also acquire and transmit, but at least one of them was pretty evident to me that it was the vector of this phytoplasma. This is just to show my computer stopped connecting to the internet for some reason, so I couldn't put the pictures of our transmission experiments we did in the field this trip. I didn't get a lot, by the way, because I was sick and almost worthless. So, like, without Erica and Melody, there'd be very little data from this first part of the trip, but this is from using Hyplaxius crudus in Florida when we did our transmission work on lethal bronzing. And this shows, excuse me, what we do is we put like a 10% sucrose into the cap of a microcentrifuge tube, cover it with parafilm, and then we let the insect feed on that. And then we test that sucrose for the presence of phytoplasma to see if it transmits. So that's just to show more or less what it looks like. And this year when we went out, we had a few objectives, and one of them was to do field transmission trials with Nisomindis from this site in Mampocony. like, if I had been feeling good, it would have been very frustrating because we didn't find a lot at this site. We only got 2 individuals. So it was much lower than I anticipated. So we really didn't get a good sampling for the transmission assays on this trip. We got 2, and I'm not sure that they fed, but sometimes that's the nature of research. So it's possible that what we got last year was a transient population and it's slowly declining in this urban area. I don't know. But that was one of our goals, was to do some transmission experiments, which was, technically we did them, but it wasn't a great turnout. Another objective was evaluating the Bismarck palms in this area, because what was evident to me was that the coconut palms were dying, but the Bismarck palms appeared not to be symptomatic or dying from the infection. So I'm curious if they're resistant to the phytoplasma, and we want to evaluate if in a site where there was lots of specimens with phytoplasma, are these palms just resisting infection, or we took a bunch of samples and we want to test them to see if the Bismarck palms can actually harbor the phytoplasma and serve as a reservoir of infection of the coconut. So this was successful. Erica got a lot of good samples. So when we get back, we're going to test these palms for the presence of phytoplasma. Another one, which we were halfway successful on, was surveying the grasses. Hyplaxis crudis in this group of succeeds, generally the adults feed on the palms, but the nymphs develop in grasses. So we were curious about finding the nymph habitat of Nisomindis here in Madagascar to identify which plant the nymphs were developing on, but also seeing if those grasses could harbor the phytoplasma and also be another reservoir. So we didn't find any in the city, but when we were in the natural areas where there's a bunch of grass around the palms, I was feeling better, so I was a little bit more efficient. I didn't find any nymphs in the grass, but we did sample a bunch of the grass. So at least we can detect or test these grasses to see if there's phytoplasma present in these natural areas. So that was our focus on this trip because of our findings from last year. So if I come back next year, I'll have some data on this to present to everybody. In the future, this project has kind of expanded over time, and as we get new data, new questions arise. So now, there's a few things we want to understand is the feeding behavior of the adults of this insect on the palms. And these 3 things are going to be lumped into a larger proposal. We're going to be trying to get funding to continue doing this research. But looking at the feeding behavior of the insects on the palms themselves, but also analyzing the genome of Bismarck palms, because if they may not be reservoirs, but they're certainly resistant to the infection as far as I can tell. So we want to try and figure out what molecular pathway allows for these palms to be resistant to phytoplasma infection. So that's kind of an interesting question. But also I mentioned before that there is phytoplasma, a similar strain in mainland Africa, in Tanzania and Mozambique. And from the data from Madagascar and what we have in Florida, I would bet a year's salary that there is a succeed plant hopper somewhat close to Nisomindis that's in Africa transmitting the pathogen. So this larger project is going to be focused on understanding the evolution of this pathosystem, where what it appears is before the continent split up, the system evolved and has subsequently been evolving in isolation in Madagascar, mainland Africa, and in the New World. So there's a there's a lot more interesting things to study about this pathosystem that we're hoping to explore in the future. And the reason Fabian could not join us is he's still stuck in Katsepe on the boat. So he fortunately couldn't join us, but just a quick thanks to everybody who's taken part in the work, to Hiri for joining us on this expedition. Last year, Miangali joined us and was helpful in the field with collecting. So if this is interesting to anybody, if we come back next year, we'll be doing similar research. So with that, I'll be happy to take any questions.